Hello. For those of you that don't know me, I am Dr. Tora Smulders-Srinivasan, and I will be taking you through, well, I guess you've already seen the week 11 lecture content, and today I'll be taking you through the week 12 lecture content, the details of the genetics of Alzheimer's and Parkinson's disease. I say details, but there's a lot of information there, as you'll see, so it is a basic overview. For those of you who haven't met me before, I'm the course leader for the MSc Advanced Biomedical Science, and I teach a number of modules on the undergraduate biomedical science degree, and teach on a couple of modules for the masters as well. So, as I said, today is going to be about Alzheimer's and Parkinson's disease genetics, and we will start off with covering Alzheimer's disease. Alzheimer's disease actually has a strong genetic component. This might be surprising if you know anything about Alzheimer's disease, because the vast majority of Alzheimer's is late onset sporadic, so does not have a single gene that is mutated to cause it. However, if you look here, it has been found that heritability in Alzheimer's disease is about 70%. So what does that mean? Essentially, that means that even in the late onset sporadic cases, there are some genetic risk factors, so certain changes in the genome that are outside of the main genes that will, when mutated, will cause Alzheimer's. So there are a lot of genetic risk factors that each one might only add a very small effect, but together it's when people have a lot of these different sort of risk factors that don't each add their own a huge amount. So it's not like the monogenic mutations. So there are some mutations, which we'll go into in a bit, that cause Alzheimer's when they're mutant, individual genes. This is an indication that there are a lot of other genes that have an impact on whether someone will get Alzheimer's or not, even in the late onset sporadic cases. So there are actually more than 40 genes or loci associated with AD risk. And one of the really interesting things about the genetics is that the genetics really shed new light on the pathophysiology. So I'm not going into very much detail about what Alzheimer's disease is, because I assume you've been paying attention in the modules so far. So Ahmed has covered Alzheimer's disease and Parkinson's disease from very much the pathophysiology point of view. So, what happens to patients, what do their brains look like, etc. And so I haven't really introduced the disorders at all, but I'm assuming that you've paid attention to Ahmed. And so the pathophysiology of Alzheimer's, if you look over here, is having these amyloid plaques that are outside of the neurons and neurofibrillary tangles, which are on the inside of the neurons. And so that's, important to know and more important to understand, but it's also interesting to see, it's also possibly vital to treating the disease to understand how these plaques and tangles actually arise, because if we can understand what causes them to arise, what caused the plaques, what causes the tangles, if we can understand the causes, then hopefully that means we can have very targeted treatments for the disease. So looking at Alzheimer's disease, actually people considered Alzheimer's to have some kind of genetic basis since the 1930s, actually. But it was only in the 1980s that systemic linkage experiments were done, and the main genes that were identified were APP, which we'll go into a lot more detail about, and presenilin, which are these PSEN1 and PSEN2 genes. And it's actually the discovery of these mutations here that allowed for the amyloid cascade hypothesis, and whether it's completely true or not, we'll go into as well. But those genetic findings are what really originated the amyloid cascade hypothesis, which, whether it's true or not, has really had a huge effect on how people think about Alzheimer's for a long time. In 1993, they discovered that ApoE has a, particularly one of the alleles of ApoE is detrimental. So if people have 1 or 2 copies of the allele, they have a much higher risk of developing Alzheimer's disease, except in African populations. So as with all of genetics, a lot of the prevalence of different mutations, of different varieties of genes, different alleles, they'll be spread differently in different populations, because it's only been fairly recently in the history of humans that we've traveled around and had and mixed our genes with people who aren't necessarily people who are in the immediate vicinity, essentially. So what that means is different populations or ethnic groups will have more of some alleles of genes and less of others, and that'll vary based on where the populations are in the world. It's not because there's any essential differences between us as humans or races, it's just because of who's actually interacted with each other over time and who's married and produced children, essentially. I mean, I guess marriage is not necessary, but which people have actually produced children together. So certainly as time progresses, there will be fewer and fewer sort of population-based differences, because I think a lot of people are, well, maybe it's just me, but marrying outside of their own ethnic populations. And then in the sort of late 2000s, so I think around 2008 or so, people started to doing GWAS. If you don't know what GWAS is, I think it stands for, let me see, genetic, I should have written this down. I mean, I know what they are, but I'm trying to remember what they stand for. Genetic something association study. Genome-wide association study. And so what they do is they basically use SNPs, which I'm not going to go into detail about, but what they do is they look at the entire genome of people, so that's why it's called genome-wide, and they look at people who have the disorder and people that don't have the disorder and try to associate different sections of the genome with the disorder and without, if that makes sense. So they'll say, these people who have the disorder have this particular SNP more often, while these people who don't have the disorder have that SNP less often. And so by doing so, what you can find are these genetic risk factors. And so those are looking throughout the genome. And again, they aren't mutations. They aren't individual genes where when they're mutated, they cause the disease. They are the risk factors, the background risk to getting a particular disease. So first, we're going to go through what the amyloid cascade hypothesis is, and then we'll work our way through the rest. So what is beta amyloid in Alzheimer's disease? So amyloid beta or beta amyloid is the most common senile plaques component. What are the senile plaques? They're extracellular deposits. A beta is a peptide, so it's made of a string of from about 36 to 43 amino acids. And it's created by the amyloid precursor protein first being translated, and then it's proteolytically cleaved. So you have the full APP protein, and then it's cut into the A beta peptide. In familial AD, which is abbreviated as FAD, and if you see it, SAD would be sporadic AD, so that's when it's not usually a single gene. So familial AD mutations will often increase the expression of the A beta 42 isoform. So I said it could be anywhere from 36 to 43 amino acids. In familial AD particularly, it's thought that maybe this presenilin mutations do this, that you get more of the A beta 42 isoform and you get more of the A beta aggregates, because they think that maybe the A beta 42 isoform is the one that aggregates more easily. So what does this look like? Essentially, if you look at the whole APP gene, there's a part of it within, I guess it's towards the C terminus, that is the A beta peptide. And so essentially what the amyloid cascade hypothesis is that amyloid beta or A beta protein or peptide is what causes the pathology in Alzheimer's disease. And this comes up because A beta protein is one of the main components of plaques. And then the idea of this hypothesis, which was very new at the time, was that the neurofibrillary NFT, cell loss, and vascular damage and dementia are all but they're all a consequence of the plaques. So essentially, they're putting the A beta first, and then making the plaques is what causes all of the rest of the disease. And this was fairly new at the time. I mean, a lot of different people came to the idea around the same time, but that's the hypothesis itself. And so what can happen is normally this APP protein is cut by a secretase, and that cuts right within the peptide. And so this part goes off and does something, and this part goes off and does something, but none of those things are pathogenic. However, sometimes when the APP goes through a lysosomal pathway, it can be cut by a different secretase, and that can cut again, and it can also be cut again here, and so the A beta peptide comes loose, and that amyloid will then cause plaques. That A beta peptide will then aggregate, cause plaques, and then the thought is that leads to the NFTs and to the cell death. So in case that figure was a little bit unclear for you, here's another figure just showing this in a little bit more detail, and I thought it was a bit easier to understand. So under normal conditions, you get more of this happening. So this is the APP in full, and it's a membrane protein, so it's within the membrane. And normally, the alpha secretase cuts it here, and you get this SAPP alpha peptide protein, which is soluble. That's what the S stands for. And that goes and possibly does things. And then you get this bit here, which might go and possibly do things. Now, in pathogenic conditions, what is thought to happen is then you get beta secretase cutting the APP, and so you get this soluble APP beta piece, and then a gamma secretase cuts it here, and that's what produces the A beta peptide. The A beta aggregates, oligomerizes, and then forms the senile plaques. And this AICD portion goes off and does other things as well. So how was this amyloid cascade hypothesis formulated? Well, one of the first pieces of information was that A beta was extracted from essentially the plaques in the postmortem AD brains. So they extracted the plaques and they looked at them. So they extracted the plaques from postmortem brains from the cerebral blood vessels and the brain parenchyma. And then when they looked at what was in the plaques, they found primarily A beta. They sequenced what was within that A beta, and that led to the identification and the sequencing of APP, which is the amyloid precursor protein. And so they found the gene. You have to remember this was in sort of ancient times, well, not ancient times, but, a long time ago, and the human genome wasn't sequenced yet. And so a lot of genes were not sequenced, and so we didn't really know what the genes looked like. So often, people would try to find genes. And so this is how the APP gene was found based on the protein amino acid sequence, and the gene itself was sequenced. And then by genetics, what they found, and so this is around the time on the timeline where I was telling you that the APP mutation was identified. So that essentially was identified in patients with early onset AD. And by doing linkage analysis, they were able to find that the people who had early onset AD in a particular family had a mutation in the APP gene. Interestingly, Down syndrome patients, when they get older, also have a higher risk of developing Alzheimer's disease. And so the thought was that that part of chromosome 21 that is extra in Down syndrome patients has a risk factor for AD. And lo and behold, the APP gene is found on that part of chromosome 21. And in the meantime, a lot of familial AD has been characterized in patients. So again, it's only 1% of all the AD patients, but that 1% is enough that, clinicians and doctors and geneticists will try to find out, well, what causes that? Because if it's a familial case, that means it's inherited within the family. So there may be a particular gene that's gone wrong. And so all the familial cases, as far as I know, are caused by hundreds of mutations in the APP gene, and then in PSEN1 and PSEN2, which all affect A beta. So you can see there was a lot of reason for saying, yeah, it is the A beta that might be the cause of all of the pathology in Alzheimer's. And so, I don't know, I found this quite hard, so I was like, oh, this is a great figure. It shows, again, exactly what's happening. So this is, again, the full APP protein. If you have alpha secretase cleavage, you get the soluble APP, and this is kind of the normal non-pathogenic pathway. Now, if you cleave with the beta secretase and the gamma secretase, so here it's the alpha and then the gamma, and here it's the beta and then the gamma, and so that gives you the different fragment, which is the amyloid fragment. The amyloid fragment will then create oligomers, so it'll aggregate, oligomers will be made, amyloid beta sheets will be made, and then fibrils will join in to make the amyloid plaque. So what are the problems with the amyloid cascade hypothesis? Oh, and I should say that presenilin 1 and 2 are part of the secretase, so that's where they get involved. So what are the problems with the amyloid cascade hypothesis? Because you may know that it's now not very sure exactly what's going on with the A beta. And one of the really key findings that has led us to wonder if this hypothesis is true is that the levels of A beta or plaques in the brain don't actually go with the severity of the Alzheimer's, the cognitive impairment, or the progression. So if A beta is supposed to be causing all of this, how come you don't get more severe disease with more A beta or more plaques? Also, when they looked in more detail, older patients who died who were cognitively healthy, they died of other reasons, they can actually have extensive A beta pathology. So if you look at age-matched controls, essentially, you get the brains from the AD patients and you get brains from older patients that also died at around the same age. They can actually have a lot of A beta and plaques in their brain, but they haven't been affected by AD. In addition, the A beta deposition happens like 15 to 20 years before the disease symptoms. So how does that work? But I think the main thing that is really caused us to reevaluate this hypothesis is that clinical trials with drugs that target the A beta pathway and clear the A beta from the brains of the patients don't actually help the symptoms in the patients. So, I mean, that's really where the problems lie. We think that A beta is causing it. All right, so we find some drugs that can bind A beta, that can clear A beta, that can clear the plaques, and then give those drugs to the patients all through the proper, clinical trial process, testing, and there's no real improvement in symptoms, and there's no, you might think, okay, well, their brains are already damaged, that's why they're identified as patients, but they don't really sort of decrease the progression through the disease either, so they don't hold them still in that sense. They're not pausing the progression either. And in some cases, they stopped the clinical trials because the patients actually got worse. So no A beta targeted clinical trials have shown effectiveness in patients. And I don't just mean the ones where they're cleaning A beta, but all along the A-beta pathway. So, like I said, where do they actually target these drugs? So this again is another diagram of what's happening with A-beta. And like I was saying, the gamma secretase, that is where the presenilin genes are. So there are 2 presenilins in this complex, I believe. And then the beta secretase is this one here. And so, and this is the APP protein, the full protein. And so the drugs that have been tried are the beta secretase inhibitors. So they're like, well, if we don't cut it, you can't get A-beta. And they've also used gamma secretase inhibitors and modulators. So again, they're saying if you don't cut the A-beta out of the APP molecule, APP protein, there's no way that, you can get the plaques developing. They've got some, so inhibitors here and modulators for gamma secretase. They've tried, I think these are antibodies that bind and, take away the monomers, the oligomers, things that attach to the protofibrils, that attach to the A-beta fibrils themselves, A-beta aggregation inhibitors, but anywhere they might actually see a decrease of A-beta along the process here, but it just doesn't help the patients. And that's where the problems with the amyloid cascade hypothesis are really the strongest, because it's, these hypotheses and understanding the disease, the main purpose is to be able to design treatments for the patients. And if these treatments don't work based on our understanding, maybe our understanding is not quite there enough. So there are other AD hypotheses, but I'm going to stop this video here. So we have more shorter videos, and then I will pick up back here in the next video. Thank you.